So welcome to our seminar of mechanics. My pleasure to announce that the speaker is Dr. Leonardo Medrano from the Technical University in Germany, in Dresden. He came to us from Dresden by train. It was so good. And the title of his presentation is very inspiring, quantum-inspired artificial intelligence strategies for molecular innovation. Okay, so as I said, he is our guest, but most the guest of Dr. Adolfo Poma, and I will ask Adolfo to introduce our speaker in more detail.
Thank you. Let me briefly say a few words about Leonardo. Leonardo is graduated from the National University San Marcos in Lima, Peru, with mention in condensed matter physics. So he has got a PhD in 2018 in mechanical engineering at the Technical University in Dresden. Then he did a postdoctoral stay at the University of Luxembourg with Professor Alexander Tkachenko, another person well known in the AI community of quantum chemistry. From 2024, he's been a research associate at the Technical University of Dresden. Something interesting about his activities in 2025, he organized two congresses, one called Sustainable Machine Learning at the Max Planck Institute for the Physics of Complex Systems in Dresden. And also, he recently came back from Peru also for the Latin American Conference on Nanotechnology that was actually in Cusco, Peru. So, and just to end this, so two of his recent articles in Nature Communication is extending quantum mechanical benchmark accuracies to biological ligand pocket interactions, and another one in the Journal of American Chemical Society, also well-known journal, in molecular simulations with a pre-trained neural network and universal pairwise force field. So we welcome you, Leonardo, and we are happy to see what you are going to talk.
So thank you for the very long and nice introductions. So, I mean, everyone, so thanks for coming to my seminar. So I will try to introduce all the topics that we are working, combining quantum mechanics with machine learning. But, I mean, this is a very multidisciplinary topic area. So we need to know a bit of physics, chemistry, biology, and also machine learning or data science. So I hope you are familiar with the topic. So, but if not, I mean, just raise a hand, and I'm very happy to answer any questions. I mean, we have been working on different topics like exploration of chemical spaces, also RNA simulation, protein-ligand interactions, biosensors, and recently we're introducing our topics in the field of accurate predictions and also molecular design. So there are different topics that I will discuss, but I selected only 3 of them, but so we can discuss more later. This is part. Okay. Okay, now it's working. So I will start the presentation with a topic that I particularly like because I have been working this in the last 4 years. It's like how to leverage quantum data to advance machine learning simulations for biomolecules. I mean, what everyone wants in this field is to compute physical chemical properties, particularly thermodynamic and structural properties, in less computational time while keeping the same accuracy as quantum mechanical methods. However, when we go from small systems to large systems, then the complexity increases by a lot because the degree of freedom also increases. Then the most accurate method that we use to study the properties of small systems, they cannot be used for large systems, and we need to use approximations like classical force-field functions. Then, since we want to keep as accurate as possible our methodologies, we try to solve Schrödinger equations for systems where we know like we can handle with the computers we currently have. And then we try to understand the potential energy surface of these systems. So what does it mean, the potential surface? So once you know the potential surface, you know like, which structure is associated with which energies, and also you can study a transition states between one conformer to the other conformer. So this potential surface describes only the energetic landscape of the system that you are interested in. So imagine this is for one case, but in our goal is to study more than one molecule. So it means that we need to compute or to perform these calculations for millions of molecules. However, so if we start doing this for, let's say, more than 1,000 molecules, then the computational resources, of course, will explode. So you need to use millions of CPU hours, also GPU hours. That's why here comes machine learning. So what we want to use is, for instance, kernel or neural network methods to learn this potential surface for specific systems. Then, once we learn this potential surface, which was computed with quantum mechanical methods, then our model will be able, we hope, our model will be able to extrapolate to systems like that were not seen during the training. So this is what we expect always from the machine learning model. However, something like many people don't pay so much attention is the analysis of the model like you are performing or rather developing. So your model should be able to predict very accurately intramolecular interactions between the atoms in the molecule. Also, very important for molecular modeling, you need to predict intermolecular interactions like electrostatics or van der Waals dispersion. And something important that maybe you know from your work in this institute is the interaction with the chemical environment. Because once you start reaching scales of thousands of molecules or you start studying relevant biosystems, then the interaction with the environment is quite relevant, especially with water. Then keep this in mind, so you need to find a methodology which is able to develop a very accurate machine learning model to predict the potential energy surface, but also should be able to predict accurately these interactions. If you don't predict accurately these interactions, then your model is incorrect. So then going this direction, in the last maybe 5 or 6 years, there has been a lot of developments in quantum mechanical datasets. In our case, 5 years ago, we were one of the first ones who developed quantum mechanical dataset for small molecules together with one group in Argonne Institute or Argonne Laboratories. So we developed this Q7X dataset, which contains 4.2 millions of structures of small molecules up to 7 heavy atoms. And then what we say is that, okay, we want equilibrium, non-equilibrium geometries of these small molecules because we want to explore all the potential energy surface of the molecules. But on top of that, so what we did was to compute 42 physical-chemical properties of these 4 millions of molecules. And this was done at a very high level of accuracy. So, I mean, as you can see now, so we're talking about billions of the FT calculation or quantum mechanical calculations at high level of accuracy. Then this was not possible in university supercomputer, and we had to apply to some US facilities and then run calculation of that. So this was the first data set that we generated in our group when I was working with Professor Alexander Chen in Luxembourg. But then we said, okay, so we study small molecules, but we also want to go to large systems, because our goal is to study proteins, also protein ligand reactions. So that's why, together with one group in Janssen Pharmaceutical, we started developing the aquamarine dataset, which consists in molecules of conformers of Gram-like molecules, especially with a number of atoms around 60 or 70 atoms. So when you increase the size of the molecule, then the molecules start becoming more flexible, and then you don't know exactly which one is the conformation with more stability. That's why you have to explore the using metadynamic sampling, we have to explore the potential surface of the molecules. And here, since you can study many conformants per given molecule, we have to define some methods to select which conformants were more relevant for our purposes. Also, besides studying the influence of the conformational sampling, we also study how the solvation improve or affect the properties or the quantum mechanical properties for these molecules. Then we build two different data sets. So one data set in gas phase, so no interaction with the solvent, and another data set with the interaction in solvent. So then this data set now is being used to develop some machine learning force fields. But then we said, okay, so now we have the small, large molecules, but we also need the connection with the valuable molecules. So then together with Oliver Runke and Professor Klaus Müller from TU Berlin, so we started developing these big data sets called GEMS data sets using two different approaches, the top-down and the bottom-up. So the top-down approach, so we take a big valuable molecule, and then we start cutting the valuable molecule into small parts like spheres, and then we run quantum mechanical calculations for these small portions. And the bottom-up approach, so we start building molecules one by one. So one atom by one atom up to 9 heavy atoms. And then we generate equilibrium and non-equilibrium conformation for these molecules, and we run quantum mechanical calculations, and then we have a big data set of small fragments of molecules and also singular molecules. And then we train the machine learning force field to study molecular systems. If you want to know more about this work, so please see the reference here, also here. Then, thanks to all these data sets that we were generating in the last years, also with the data set from other groups, there has been a lot of developments in machine learning methodologies. This is a figure from a review paper that we wrote together with Adolfo this year. So here we split the methods of the machine learning methods in 4 blocks. We have the hybrid machine learning molecular mechanics, we have equivalent neural networks, we have machine learning augmented semi-empirical methods, and kernel methods. As you can see, there are already methods that you can use to study molecules in solvation. So like a solar, grappa, or whatever here. So we're talking around 1 million, 10 million of atoms that you can study using machine learning force fields. However, there is a trick here. So this, to study this system, you really need very good GPUs to run simulations. So if you don't have access to GPUs or to the current state of the art of GPUs, then we stay in this range here. So these simulations, they are being run mainly in NVIDIA GPUs, computer. So this collaboration with some companies. But then here I want to talk about what we did in terms of inside our academia network. So how we use equivalent networks to make the simulations of molecules. So then again, in collaboration between the University of Luxembourg, TU Berlin, and Google DeepMind, we developed a new architecture, which is called Socrates. And then to train, once we trained Socrates in 10 different data sets. This is a data set I showed you before, but then we did a pre-selection of which molecules were more relevant to train this model. And now here, so let's say comparing to the previous state of the art. So we are splitting the potential energy in different terms. So we have a short range term, we have a semi-local, and a long range. So this long range in particular is something new in this method or in this work, because now we're trying to tune how electrostatic and Van der Waals dispersion are predicted in the machine learning model. So as we said before, in our case, we try to introduce more quantum mechanical features to the development of machine learning methods. However, if we split the potential energies of the systems in different components, then we also have to improve or tell the network to optimize the parameters, having this information separate. That's why we have 3 different loss functions. One is related to the atomic forces, which involve these two terms, and the other ones which relates to the dipole moment and the huge wave ratio. So these two terms are related to the long-range components of the potential and energy. So then, yes.
You said that we can interact. So explain in a little bit detail how you construct these loss functions and because I understand that the aim is to find the multidimensional potential, right?
Mm-hmm.
And so you are like having the potential, you are using, I understand, some gradient descent, right? And to minimize loss function and simultaneously, I mean, to do it, you are performing molecular dynamic simulations that probe this potential.
In this case, no, because in this case, we're using the data set. In this data set, you are going to contain atomic forces, dipole moments, also energies, and so on. So what we do here is like, so this one here is the prediction of the network. So we are trying to match what the network is predicting to the real value that we have in the data set.
So in the data set you have the potential.
Yes.
Okay.
So in this data set, you already have all the information of 4 millions of molecules, like energies, forces, dipole moments, and atomic charge.
Okay. So you have like structures associated with potentials, and this is the training set. Okay, and part of the training set is a validation set.
Mm-hmm.
Yeah, and then you expect that you train your network that will be able to predict potential for other molecules. Yeah,
Yeah, that's the point.
Yeah, but how these other molecules are set, because here you have structure. Yeah?
Yes, here we have like a small and large systems. So and then what we are expecting is like a network will learn to interpolate and extrapolate to reals that you don't have in your data sets. So that's what we are. I mean, that's what we are generating this machine learning models.
But in the training set, you have the structures or you have just sequences?
It's structures.
So it's structures and potentials. Yeah? But then to use it, yeah, you will have to use it without having structure. Right?
Once I can show in the next slide. So I mean, once the model is trained, so we have the potential. So then you can introduce any of the systems that you want on the initial structure, and then the potential will just evolve over time. So you need one initial condition, so which is one initial configuration. So, and then this initial configuration will just evolve over time.
Okay, so you will then start from initial configuration. You will, by your machine learning, you will get potential, and it will tell you how evolve the structure, how you make, in a sense, one step.
Exactly.
And then you will get, yeah. So okay. So one more question. But if you are training on these structures you have for training are sort of equilibrium structures?
No, they are equilibrium and non-equilibrium.
Okay.
They are both, so we have more information.
Okay.
So then in this world you can see like we have applied this new model which is called solar. I mean, you will understand why in the paper. So then we apply already to lipids, proteins, carbohydrates, and some nucleic acids. So, I mean, this is very interesting. So we are pushing. So our model was not the first like being transferable in different biomolecules. But I mean, there are still some limitations. We were discussing. So if you go far from the training set, then the models start failing because, especially when you have like a biomolecule, they suffer like a very big structural transitions like RNA and so on. So then the model cannot capture this because we don't have this information in the training set. Since these molecules or these biomolecules, they are a bit rigid, so then we can describe correctly them. So, but I mean, if you go like biomolecules that are more flexible and they solve some transitions, then there are still some issues to solve there. So that's why, together with Adolfo and also Simón Poblete from San Sebastián University in Chile, we said, okay, so in parallel, let's start developing a new machine learning forklift, but only focusing on RNA simulations. Because I mean, RNA, as you may know, so is very interesting and also quite complex to study. So I mean, many people are developing classical forklifts to understand what happens in some RNA structures. And maybe some of you know this representation, but in our case, since we're interested in atomic interactions, so we have to, I mean, use the whole atom representation. But since to study this big system which contains billions of atoms is very hard for quantum mechanics, then we decided to start analyzing or to start thinking how to develop a more transferable and scalable machine learning potential, only studying the adenine binding. So for this, as every machine learning study, we have to generate first the quantum mechanical datasets. So we, to generate the geometries in equilibrium and out of equilibrium, so we run vertical chain molecular dynamic simulations using classical forklift, and we found that this adenine dimer contains 6 different, or exists in 6 different clusters. So you have the A4 or stacking configurations here, but also you have the unstacked configuration, which is clear because to go from one stacking to the other stacking configuration, sometimes you need to go through an unstacked configuration, right? So then we have now this structural data set which was computed with classical forklifts, but since we want quantum mechanical accuracy, then we run single-level calculations using 2 different quantum mechanical methods. So we use semi-empirical methods which are faster to compute, and also we use first principle method which takes a bit longer to compute, but they are more accurate, right? So then we have the energies, or the description of the potential energy sources for all these structures that we generated previously. And here we found like DFT, or first principles, give a larger, fluctuations of energies compared to a semi-empirical method. This is kind of expected because semi-empirical methods, they are an approximation to first principles, and they sometimes fail to describe long-range interactions. So that's why maybe what we think what's happening here is like as an empirical methods are describing maybe local interactions in the RNA system, but DFT or first principle methods, they are describing correctly everything, local and long-range interactions. Here you can see, if you analyze the advanced dispersion energy or minimal dispersion energy, also the fluctuations for DFT, they are larger compared to semi-empirical methods. So I mean, our explanation is going in that direction, but I think we need to discuss more. I mean, that's why I came here to discuss more about this work with Adolfo. And also something interesting that we found was the following. So, I mean, I don't know if you're familiar with molecular simulations, but in classical force fields, what people or physics they use is fixed atomic charge. But here, if you analyze the atomic charge evolution during the replica chain simulations, you can see that this is the standard deviation. There are some fluctuations in the charge. So it will go from 0.01 to 0.03, which in some cases is 10% of the fluctuation of the atomic charge. So it means that if you consider fixed atomic charge during the simulation, that's wrong from the quantum mechanical point of view. So in this way, using these data sets, we're trying to inform our machine learning model like correct this atomic charge during the simulation, which is also introduced. This information is introduced in the energy and the atomic force, right? So just some preliminary results that we got. So I mean, this is the reference data. and the population in each cluster. And this is our results using a machine learning potential training the datasets I presented before. So S means how much stacking we found in the molecular dynamics trajectories. And as you can see, our models, they don't go more than 40% of stacking here. Any of our models goes beyond 40%, which is a bit disappointing, but I mean, makes sense, no? Because to go from one state to the other state, or from one cluster to another cluster in these RNA simulations is quite difficult, so it's not so trivial. No? And we said, okay, so this is what we generate, but what about if we compare with the state-of-the-art foundational or generalizable machine learning workflows? You know? One of these is Solar, which was trained by, previously by my previous group. So, the other one is Maze-Off, which was trained by the group in Cambridge. So these two are currently the state-of-the-art models in biomolecular simulations assisted by AI. And here with Solar, we found 86% of stacking, so which is more than what we got with our trained models. But with Maze-Off, we found 30%, which is more, but something critical in the results obtained with Maze-Off was like we don't find the A4, so this orange circle, which corresponds to the A4, we don't find it here. So which means like this potential never visit this cluster, which is wrong, because in this type of dimers, so adenine dimers, the most stable cluster is the A4. So this already tells you like this model is not good and so on. So this is, I mean, we have to do more analysis, but here what we are trying to do is trying to understand if the current state machine learning workflows they are able to predict these structural transitions even for simple systems like a one adenine dimer. So, okay, so I mean, going far from the equivariant neural networks, another topic that I really like is this machine learning augmented semi-empirical methods. Why? Because in these methods, so in this technique, so we can introduce more physics to the machine learning model. You know? So if you are not familiar with empirical methods, so one of them is the FTV method, or density functional diagrammatic methods, and it has been applied to different kinds of studies like self-assembly monolayers, biomolecules in water, molecular crystals, and molecular electronics. So the FTV total energy, as a typical empirical method, can be split in many components. And then, where the three first components they are related to the electronic interactions, while the last component, which in principle will contain all the information that you don't have in the electronic components, is called the repulsive term. And in the standard FTV, this repulsive term is fit to atomic pairwise potentials. So it means that it's only considered like a two-body potentials, which bring problems to the transferability for the FTV. So that's why every time that you want to use the FTV for new systems, you have to fine-tune the potentials, this repulsive potential, and try to optimize it for the application that you want to study. In our case, 5 years ago, we said, okay, but this is not possible. How we have to tune this potential every time that we want to study something new? So that's why we said, okay, then let's create, or let's develop a machine learning model capable to be transferable to any kind of systems, and also small and large systems, dimers, and so on. And our first work, in our first work, we used this deep tensor neural network to learn the correction from DFT data to the electronic DFT. So in this case, we don't touch the electronic components because we believe they are correct, so they were correctly parameterized. But the repulsive is the problem here. So that's why we're trying to improve this repulsive by training the machine learning potential. So in this work, so 5 years ago, we found by training this potential, so we were able to improve different properties like optimization energy, stability ranking, customization, and so on, even rotational profiles, and you can see here, so our model is the red one, and the reference is the black. However, one of the problems this work was like we were able to go to systems larger than heavy atoms, which is critical if you want to study molecules, and also the long-range effects were not described correctly. So our model was working perfectly for systems from 1 to 7 heavy atoms, but then it was not able to extrapolate to large systems. That's why in the last 2 or 3 years we were trying to find a way to optimize this model and make it more transferable for large systems and especially for molecular diagnostics because the target is to study protein-linear interactions. So then we started using equivalent neural networks, and now we have these many body delta TD potentials which were trained in Q7X dataset, and also we test some models trained on tiny datasets here. And now, as you can see here in these 2 plots, our models, all of them, they are closer to the reference data for small molecules, and even that they can predict better than the responsive potential, like the standard responsive potential in the FTV. No? So we can predict better the long-range correlations in small molecules. But okay, we say, okay, small molecules, we improve it, but okay, where is the challenge, the real challenge? No? So then we go to molecular diagnostics. So in this case, I will present some results where we didn't use molecular diagnostics for training the machine learning potential. So we were hoping that by learning only small monomers, we were able to extrapolate to molecular diagnostics. And this is what we found. So here is the distribution of the error in predicting the interaction energy between molecular diagnostics. And these are the results for the standard DFTB3 for XTB. And this is our results, our best results here. And this is the result using a standard machine learning potential. So, and as you can see here, so our model is performing better than the standard time-bounding models, and also better than the machine learning potential, since the distribution of the errors is quite narrow. And if we compare to our model like was developed 5 years ago, so you can see clearly the difference, right? So it's this one compared to this one. So then it means that we were able already to improve by a lot the prediction of non-covalent systems. No? So then I call this model EquiDTB for equivalent high-fidelity. And then we also predict atomic forces, since we want to study molecular dynamic simulations of molecular diagnostics. So let's see how the model predicts atomic forces. You know? Our model is the yellow dots. And you can see in all the plots, so we get better accuracy than the reference machine learning models. Also, if you want to see some numbers, so these are the errors for the relative error of interaction energies and the errors for predicting the atomic forces, which is by far better than this standard DFT. So then we said, okay, now we can predict non-covalent systems, but let's go to larger systems, no? So let's go to something more relevant for pharmaceuticals, also for us. Then we study sampling gas, and we study the legal to Q5K, and we run molecular dynamics with our models, and also we run molecular dynamics using the reference models. No? And as you can see here again, so we analyze the prediction forces. Our model is much better than the other ones. You know? So it means like by only training our model with small molecules up to 7 heavy atoms, we were able to study the properties or the properties of larger systems and also non-covalent systems. And then we said, okay, now we can predict energies and forces, but what about non-rational properties? Then we analyzed the set of alpha amino acids, and we compute the non-rational modes for all these amino acids. And as you can see already here, so our model in red here, so this triangle, so this is smaller than the time-bounding methods here. And also we compare to the reference machine learning potential. So our model is producing much better results than this reference potential. So then it means that we were able to improve by a lot the prediction of DFTB methods, and the computational cost here is only twice the standard computational cost of the typical time-bounding calculation. So it means that we have DFT accuracy, but at a much lower price. So then here, since our goal is to study protein ligands, now we are testing this model in this kind of systems. And so here I want to stop because I want to tell you since our target is these protein ligands interactions, so recently this year we developed these new datasets or new mechanical datasets where we combine large graphite molecules and small monomers and try to analyze how these how the interaction of these two components they are affected by the conformational sampling. So, because as we say, if you increase the size of the molecule, then the conformers will start making the conformational sampling is more relevant. So, because you need to understand which conformer is more energetically stable than the others. And this gets more complex when you have two molecules interacting. In this work, what we did is, if you are more interested in this type of work, so we analyzed 6, 7 reference quantum mechanical methods, like a copper cluster, diffusion Monte-Carlo, hybrid DFT functionals, double hybrid DFT functionals, and so on. So we did a very robust analysis of which functional could be more relevant to study these interatomic interactions. And of course, now we want to predict these interactions using the previous model that I developed, but this is ongoing work. So that was the first part of the talk, about the molecular simulations. But so I also want to tell you how we can use machine learning techniques for the freedom of design of molecules in the chemical space. So what does it mean that? We will see in a few minutes. So, this is the cover that we published in this journal, Chemical Science, 2 years ago. Here we propose an idea instead of analyzing the chemical space only on conformers and compositions, we have to analyze the chemical space by properties. Because, properties, we know properties depend on conformers and composition, but some properties, they are not affected by the conformational sampling, or they are not affected by modifying the cell by replacing one sulfur by phosphorus. That's why we need to understand how properties are correlated, and also how we can use this information, or how we can navigate in this property space in order to optimize the molecule for a given application. So this is what we propose in this paper. So we call it freedom of design concept, but if you are more interested, also you can do it better. So then, by using this concept of navigating in the property space, we decide, okay, let's develop a model that we can go from property space to conformal space. So then, this is called inverse design. So then, by any set of properties that you want, so you can generate molecular structure. How we can do this? By using conditional genetic models. In this case, we use one technique called variation encoder. And here, what you need to introduce is the conformational information of the molecule, here one encoder, and then also, and of course we have a loss function associated with this encoder, and then you also need to introduce the information of the quantum mechanical properties corresponding to each atomic structure. So then you will optimize this latent space here in order that this decoder will learn how to correlate the structure with the properties. So once this latent space is optimized by minimizing this loss function, you will have a model where you just introduce the properties that you want, and then at the end you will get the conformer for the molecule. So you will get a three-dimensional conformation of the molecule. So this is something like that has been developed in the last 2 years, like many people are working on this, but we are trying to optimize this method nowadays for large systems, especially for protein ligand systems. So then, just to show you how this model works, so what we did was to study the property space defined by the molecular possibility and the band-advanced interaction. And then we select 3 different targets, A, B, C, and then we select the best or the best 5 samples here. And then, something surprising here was the model was learning some physics without telling to learn the physics. Because, for instance, if you go from point C to point B, you increase the molecular possibility, which, if you are familiar with physics, means like the molecule will get larger. So, because it's an extensive property, right? Then the molecule was getting larger. So, but also, if you go from point C to point A, you are increasing the mass interaction, which means that the molecule will get more larger and then also compact. Because the many interaction will be more relevant. And what's happened here? But we didn't tell the model to learn this. So the model learned this by just learning the correlation between the properties. So this was something interesting, and then we said, okay, how is it possible? So then we define a new parameter, which is the D parameter, which tells you how much each property contributes to the generation of the molecule. And what we found was the following. So here is a clear difference between extensive and intensive property, and especially so the best property to generate molecules was the exchange correlation energy. So which, if you're familiar with quantum mechanics, this is also related to the interaction between the electrons. So then also we have the kinetic energy, which was the second place. So it means like that the model was finding some correlations between the structure and these 2 properties, which is clear because these 2 properties contain information about the valence electrons, the core electrons, and how these electrons are interacting. So that was very interesting, but also we found correlations with stability and ED and condensation. And what we found is also in our model, the intensive properties were not relevant for the prediction. So you can see here. So the relevance for genetic molecules was not so much for the intensive properties, which could be related also to the atomic representation that we use for training the model, but we can discuss that later. Then what we say is, okay, let's try to explore regions where we didn't have so much data, and then let's see if we can populate these regions. And yes, we were able to extrapolate to regions where we didn't have so much data. So then this model was able to generate molecules again up to high heavy atoms, but this limitation was because of the atomic representation. But now we have models that are using diffusion modeling that can go to larger systems. So then, to summarize this part of the work, so as I was saying before, so if you want to study or you want to apply machine learning techniques for molecular modeling, so what you need first is to generate a quantum mechanical data set. Then you need to understand the data set, because if you don't understand which data set you have, then you won't be able to define the problem that you want to study. So, because the problem will be associated with the data set that you have. You cannot expect if you have a data set for molecular dynamic simulations, that data set will be used to generate novel molecules. So that's not correlated. Then, once you define the problem, so you can define also which machine learning technique you want to use. So like a genetic model, or deep networks, or machine learning augmenting semi-empirical methods, and so on. And then, of course, you have to validate the machine learning model that you have by producing more data, or you can also validate the model using, or improve the model using active learning techniques. So this is what now we're trying to implement in our workflows, because generating data is very expensive, and especially for systems that you don't have any idea, so that you don't have so much information. So that's why you use active learning, then you can reduce the computational cost like you need for developing these machine learning models. And of course, this active learning will be assisted by these data-driven electronic structural methods, or in the best case, you have some experimental partners can be also assisted by experiments. So just to close the presentation, so all these methodologies that we are developing, using quantum mechanics, the idea is also to use this or something more relevant for different fields, like biophysics or biology. So that's why we are pushing the use of quantum mechanical descriptors to predict properties like toxicity, absorption, and so on. So this is our recent preprint where we are predicting, or we are developing machine learning models to predict toxicity and also lipophilicity. We are explaining which quantum mechanical property is more relevant for the prediction of these properties using sharp analysis. If you are more interested, so you can see here. And also, another direction that we are going is to develop datasets to understand how this digital of approach system work. So because we have many receptors, or people generating receptors, but generating receptors is also very expensive from the experimental point of view. So from using theoretical approximations, we are trying to understand how to generate the optimal receptors for specific gases. So, and that's why this year we generate this molecule data set, which contains around 10,000 systems, combining graphene with molecular receptors and also some gases. So, and now we are developing machine learning models to try to predict which receptor could work for the specific gases. So, or how to optimize the performance of the receptors. Okay, so then to conclude the presentation, so I would like to thank all these members of the who contribute to the projects that I showed in this presentation. So also the funding and also the international resources. And if you want to know more what I'm doing in TUD Dresden and with the collaborators, so just scan this code and you can go to my website, or you can send an email or contact me later and so on. And then thank you for the attention.
Thank you very much for a very inspiring and intriguing and difficult lecture. We have now time for questions. So maybe I will start by asking you, what do you mean by quantum mechanical properties of molecules? Use such a term.
Yeah, in our case, so we're trying to study or to describe a molecule based on quantum mechanical properties like molecular permeability, dipole moment. So any observable that we can compute using quantum mechanical methods. So this is the one.
Dipole moments of certain groups of atoms, yeah? Or
you can get the atomic dipole moment, or you can get also the local dipole moment.
Thank you very much for your interesting talks. I would like to ask for the case of you build the machine learning to build the fog field for the MD simulation. You see that, for example, it's improve a lot some property like charge distribution or something like this. But in the case of the general performance of system, it did accelerate the simulation process compared with a normal model like on-atom or maybe amber fog field or charm, something like this. How you compare the performance between two?
Yeah, this is a common problem that we have in machine learning community because so we, in terms of speed, so Tesla for is there faster than our models. So because I mean, our models they are not parallelizable yet. So they run single GPUs at the moment. So, but I mean, nowadays, as I say, you know, like I said, now when you discuss with someone from industry like Meta or like NVIDIA or Microsoft, so they have more resources in that direction and they're investing a lot in optimizing the methods to make it more parallelizable and so on. But in our case, I mean, most of the models that we have, they are single GPU, and they have also limitation in terms of memory because you cannot accommodate too many atoms. So there is within the say like 100,000, maybe it's a lot. So like unit GPUs like 80 gigabytes and even more. So that's why we have some hardware limitations at the moment. So, this is a problem that why just for fees they run faster. So, but I mean, hopefully also like now they are people optimizing the methods to reduce the number of parameters and also to make it more friendly with some normal GPUs like 42 gigabytes and so on. But I mean, something like we want to do also in academia is to introduce more physics to the development of machine learning algorithms. In that way, you can reduce the computational cost. So, because I mean, the problem with the big models trained millions of data points is like they contain like just 110 million of parameters, which of course slows down the simulations.
Okay, thank you very much.
We have a question from Professor Ignatsky.
What is the advantage? I think I have some intuition, but I would like to learn it from you. What is the advantage of learning from existing structures the potential, and then using this potential to predict structures in the case you don't know it, from just learning from sequence structure? Yeah? Because this is like the very brute force approach that you are learning, that you know sequences, you know structures, and you are learning the relation between structure and sequence, and then you are skipping all molecular dynamic simulations. Maybe you are leaving it for fine-tuning. Yeah? So what is the advantage?
I mean, what we want to do here is to develop a transferable and generalizable model by using the less data possible. So that's why we are improving how the network is there with this data, like long-range interaction or short-range interactions, using these terms that I was saying before. Because if you want to learn just brute force learning this pyramid, so then you will need like more millions of data points, because you need to learn how to extrapolate from output. And this is what these big companies are doing. So they are using 20 millions of data points, and they are learning from that. But in our case, in our previous model, we showed it only for millions, but you can go from small systems to large systems. So the idea is that. So the idea is to introduce more physics to the architecture of the network and try to reduce the number of data points that you would need to generate more transferable machine learning. I mean, but we don't need all these records of the dynamics. So we just need like a specific. That's why also we introduce these active learning loops, because this technique helps you to select only the data that you would need to develop a more transferable model.
Okay. In fact, I was expecting a little bit of different answer, because I thought that the advantage that you are learning the potential is that you can use your approach to study the molecules that are flexible and are working because of being flexible. Yeah? And if you are learning the structure, then I think it is easier to approach the molecules that have been crystallized or that have been intensively simulated using molecular dynamic simulations. But I understand that the competition now is to predict how molecules without fixed structure, how they are moving. Yeah? For example, in biology, yeah, the proteins that are changing conformation, they are expected to play regulatory roles, yeah, that they are flexible. So I would expect that, but maybe I'm wrong, that your approach can be like applied to finding how proteins that are switching between conformations, what is the probability distributions of these conformations. Yeah.
As I said before, that's the goal. So you want to study how this population change. But nowadays, most of the fields, since they are learning from rigid molecules, so they can now go to this kind of systems.
Yeah, that's why I asked about.
So yeah, the idea is like using these physical terms in the architecture, so you are able to extrapolate to these large or more complex structural transitions. So, but that's still ongoing. So I mean, like, no one has done anything else. So that's why I think was related now. It doesn't afford.
Okay.
Yeah. So, very simple question. So, the possibility of such an approach, having this possibility, it's important the fact that the Schrödinger equation is linear. Is it very important?
Oh, yes, I said before. So yeah, also something important in this field is like how you deal with the Schrödinger equation, like which level of theory you use. Because you can use like a very precise level of theory, like a Popper cluster or quantum Monte Carlo and so on. But to generate data sets with this level of theory, so it's not possible to go to millions of data. So that's why you need to use approximations, like DFT, or Harry functions, and so on, which somehow reduce the computational cost. But I mean, this is still very expensive. So I mean, that's why this work that we were doing, so we're trying to see if using big methods, so you can predict or you can reproduce data set at the level of DFT.
So, in your investigations, have you?
This is very, very impressive that you want to design on the molecular level for special properties, biological, physico-chemical, connected physico-chemical. But let's say elementary concept of sweet and salted. Solid is something we use every day. Could you enlarge such materials that generate some property besides known already solutions? Say poisonous, toxic, sweet, and salted. This is biological property. And could you say simulate only existing solutions so you can design new molecular solutions for such properties?
Yeah, no, that's possible. So the point is that everything depends like how you introduce your information in the model. So because you can introduce this information as an interaction with the solution or with this solvent. So in our case, we're trying to see if these interactions with the environment and also with the intramolecular interactions, they can be described in these quantum mechanical properties. But I mean, you can also use in our physics, they have some properties like experimental properties that they can be used also as a descriptor, and this can be this can extrapolate to design new influences. But this is possible, so that everything depends like how much data you have about these systems. Because unfortunately, so these machine learning models, they depend on data. So if we don't have the data, then you can extrapolate. So it's possible.
Okay, so quantum mechanics effects are let's say, only quantitative, yes? So, have you found any qualitative effect just coming from quantum mechanics?
So I mean, like in the case of molecular simulations, so we are trying to see actually, so if quantum mechanics will produce some new trajectories, for instance. You know? So I mean, this is what we are trying to discover. So, but something here in this part, for instance, something interesting is like no one has tried to correlate interactions between atoms or quantum mechanical properties with these biological responses. Because I mean, toxicity is something like you measure in the lab, and you need to run many samples and so on, but how you work or how you try to understand from quantum mechanical point of view, how the molecules behave there. And we found that it's quite interesting that some properties like homo interaction or homo lumula, so they could give some information about toxicity, but still this connection is not clear. No? So, for example, conductors, we know homo lumula, so there's integration overperformance of semiconductors, but how you connect this with biological responses.
Because, I want to ask one question about the technical problem. So, in your case, when you train that kind of huge system like this, do you use some common training algorithm, I mean platform like TensorFlow, Keras, or you have to build your customized training system for this kind of huge data? So.
In the case of the machine learning for fields, so we have our own architecture in PyTorch, which optimize the model using the dataset. So, but any dataset you can use. But I mean, everything's built on PyTorch. So.
All of this also you in Python, right?
Yes.
It's the standard library now.
Yes, yes. I mean, there are many standard libraries. Even Google has their own libraries now. So they have NVIDIA, so they developed recently like one library for molecular simulations. So that's why I said, because in academia we're trying to, so the competition is unfair with big companies because they invest more in this direction, but we're trying to introduce more physics and chemistry in that direction. So,
okay, thank you very much.
Okay. Is there no other questions? Thank you once more.
Thank you very much.